The World Health Organization has called for an urgent increase in actions to address the epidemic of non-communicable diseases such as cancer, lung and heart disease, which collectively account for 71% of all deaths globally. What have we learned from improving R&D productivity for complex chronic diseases? How can different modalities and technological innovations further improve our efficiency and effectiveness in helping patients? Can we reverse, prevent, and cure chronic diseases in the next decade? Please welcome Sir Menelaos Pangalos, EVP and President, Biopharmaceuticals R&D, AstraZeneca, and Steve Yang, Co-CEO, WuXi Healthcare.
Sir Mene, it is a great pleasure for me to welcome you to the WuXi Healthcare Forum 2022. And it is wonderful to hear your insight and catching up. Complex chronic disease. Clearly, this is something, a major theme of this year's WuXi Healthcare Forum. Let's start with clearly something that really has an impact to our industry. In the past decade, under your leadership, AstraZeneca has transformed your pipeline and approach to R&D. And what have you learned on changing productivity and mitigate risk in biopharmaceuticals?
Yeah, thanks, Stephen. I think if there was one message I would focus on, it's to focus on the quality of your pipeline, not on the quantity of your pipeline or the volume of your pipeline. If you look at our success rates as an industry, they're very low. The average now is around 7-8%. So then that's from when you have a candidate drug, you've completed, you're doing GLP tox and trying to get yourself through clinical development. So it means we're failing 92% of the time. I mean, we're very good at failing. And AstraZeneca from 2005 to 2010 was even better at failing than anybody else, because if you measured our success rate was around 4%. And so we were failing, 96% of the time. So it really was not very good. And that's with a budget we were spending, I think, about $5 billion a year. So looking at all of our data and looking at what other companies have done as well, we focused on 5 things. The first is understanding the biology of what we work on and really making sure that we work on fewer things, but in areas where we can really dig deeper into the biology and constantly try to disprove or prove our scientific hypothesis. The second was something that we call right tissue, which is around defining how much you need to inhibit your target of interest in the tissue of interest. So let's say you've got to inhibit a receptor in the heart. Show me how much I have to block it by, right? Is it 80%? Is it 90%? Is it for 6 hours? Is it for 24 hours? And demonstrate me a way that I can measure that in animals and then ultimately in people as well. And that enables you to demonstrate proof of mechanism. And that's important because when your programs fail, and as you know, as I said earlier, we fail a lot. You want to know that you failed, but you've tested your scientific hypothesis, because then at least you've learned something from your experiment.
Yep.
The third of our 5 R's was right safety. That's defining and understanding your safety signals. When you pick a target, what are the risks? How do you try and mitigate those risks early on in research? Because what we found out was that when you had a safety risk, it tended to get worse as you moved further along. It never went away. It just got worse and more expensive. Yep. And so weeding out safety risks early on saves you a lot of money and a lot of time. And then again, enables you to focus on programs that have got a better chance of being successful. The fourth of the 5 R's was right patient, defining the patient population that has the best chance of responding to your mechanism. Because if your drug doesn't work in that population, it's not going to work in a broader population. And then the 5th R was right commercial. And by right commercial, I don't mean is it going to be $1 billion revenue drug. I mean, why would anyone want to prescribe the drug, and why would anyone want to reimburse the drug? And therefore think about what experiments you have to do in the laboratory in terms of the comparators that you use, and in the clinic, in terms of the comparator use, because you have to ultimately project when your drug launches, if it's successful, what will it be being compared to? And it's not today's standard of care, it's tomorrow's standard of care. And so thinking about those comparisons through your development is, I think, very, very important. All of those things together helped us change our culture. We've gone from a 4% success rate now to we're almost up at 30%. So we're kind of one of the best performing companies in the industry. I think in 2012 to 2016, we'd got above 20%. And now if you look at our success rates, and obviously with the COVID work that we've done as well, our success rates have gone up. So we're now sort of in the sort of high 20s, low 30s, depending on the therapy area and what we're doing. So we still do very well, and we're still very good at failing. So we still fail much more than we succeed. You know, we fail 70% of the time now. But I think it's a much healthier place to be. And I think you've got to be careful not to push it too hard, because if you're not failing, you know, a good amount, you're probably not trying to innovate enough. Now, what I would say, Steve, is the reason why we fail now is very different to the reason why we failed from 2005 to 2010. If I look at the work that we did from 2012 to 2016, when we looked at the next round of our success rates, we weren't failing anymore because we had lousy chemistry. We weren't failing for safety. We weren't failing because we weren't demonstrating proof of mechanism or showing good PK/PD. We were failing because we don't understand the diseases well enough.
Yeah, it's now back to the disease, complex disease biology.
Exactly right. It's back to disease complexity.
Complex chronic disease. Clearly, this is something, a major theme of this year's WuXi Healthcare Forum. So what is your view on the future of biopharmaceutical innovation and its impact for patients, especially for this complex chronic disease?
Yeah, so it's a great question. Of course, it's great treating rare diseases, it's great treating cancer, but we're talking about thousands or a few million patients versus the billions of patients that suffer from cardiovascular disease, diabetes, lung disease, etc. I think what we're trying to do in chronic diseases is really break them down into subsets of diseases. Diseases generally are quite heterogeneous, whether you look at NASH or chronic kidney disease or asthma and COPD. And so trying to define subtypes of patients within a broad chronic disease class, either using genetics or biomarkers, is helping us try to define the right patient again for the right mechanism. And actually, when you look at the molecule that we have in development now, which is targeting a cytokine called IL-33, we're trying to define a subset of patients through their genetics and through their biomarkers that are more likely to respond to an anti-IL-33, let's say, than an anti-IL-5. And then you have other programs like tezepelumab, which is an anti-TSLP, which seems to be able to work in a broader population. But we're trying each time to define the patient population that's most likely to respond to a particular drug. But we're also investing very heavily in genomics, Steve, as you know, to sequence several million genomes, because by looking for rare variants of disease that either protect or cause disease, you can get better insights into some of the pathways that may be important in driving slightly broader, more chronic diseases. And we have, you know, many examples. Give an example, you know, ApoL1 in chronic kidney disease, right? That's a very specific polymorphism in a gene that's actually a gene that protects us from trypanosome infections. But we know that it makes people vulnerable, particularly African Americans, to chronic kidney disease, and they have a much poorer prognosis if they have some of these risk alleles. So it basically gives us an insight into a subset of patients with chronic kidney disease and a way of targeting them specifically by going after the
right patient.
It's going after the right patient, exactly right.
So with so many new insight using genomics and human genetics, you can now have precision medicine type of approach. And obviously, this also, the type of targets and the disease biology probably requires different modalities. So what's your view on the conversion across different molecules, modalities, hardwares, and software innovation for improving patient outcomes?
Yeah,
since your R&D organization, if I look at your R&D presentation, it encompasses so many different modalities and a different approach in combination.
It's a great question, Steve, and it goes back to, again, if you think about why was it important. When I first started in AstraZeneca in 2010, we were basically a small molecule and antibody group, and actually we were predominantly small molecules with antibodies starting to grow. If you look at us today, we probably have 12 or 15 different drug modalities that our scientists can use, ranging from cell therapies to antisense or oligonucleotides or siRNAs to messenger RNA to PROTACs to anti-kalins, Fab fragments, ADC. I mean, you name it. We have a lot of different platforms, and the reason we have so many different platforms is because as you start to work in more unbiased ways on understanding the complexities of the disease that we work in and ultimately the genetics of the disease that we work in, we start to come up with targets where if all you have is small molecules and antibodies, you basically have to stop because there's no way of targeting them. And we've come up now, and I think kind of it gives you a sense as to the level of innovation. We come up with targets every year where there's only one paper in the literature, and it's a paper that basically describes the gene but doesn't know what it does, and hasn't related it to the disease that we're interested in, for example. And so there, by having a variety of technologies, that it enables your scientists to still prosecute that target and that biology. So I think it's incredibly important that we have a broad set of tools that enable us to go after any target, whether it's an RNA sequence or DNA sequence, whether it's a transcription factor, whether it's a kinase, a receptor, intracellular, extracellular, whatever it happens to be. And so that becomes very, very important. The other piece that I think has changed quite dramatically, and this is really more recent over the past 5 years, is how we use artificial intelligence and high power computing to enable our scientists to make better decisions and to be more efficient and effective. And the analogy, if you just think about, I mean, I know there's lots of scientists from pharma watching this video, but think about, your favorite target or the favorite area that you work in, whether it's in heart failure or kidney disease or cancer, and you think about just one target in that space, and you think about how many papers get published every day, how many patents get published every day, how many presentations happen at conferences every week or month. And it's impossible for any single scientist to keep on top of all of that information. And that's just if they focus on, let's say, if you're working on EGFR receptors, you'll just be focusing on lung cancer. But think about all of the EGFR papers that are happening in kidney disease and heart failure and Alzheimer's disease. And so by being able to create systems and use AI to basically put all of that information that's coming out every day together into what we call now knowledge graphs, so that we can actually look at everything that comes out and put it into a format that our science can start to look at patterns and generate insights in what I like is a very unbiased way. So it doesn't have also the bias that the scientist has of this is my favorite area, or this is the disease, or this is the pathway I like. We're coming up with a lot of very novel insights that you wouldn't otherwise come into. And then you need the insight of the scientists and the chemists and the physicians to take that and use judgment as to what to work on first.
You generate new hypotheses based on
to generate new hypotheses, and then of course as you generate data, that all goes back into the knowledge graphs, and you update them, and so they're living databases and living graphs. And it's the same in chemistry. If you think about synthetic routes or designing molecules, using computers now to help you think about what the best options are, and not the easiest ones, but actually the best ones that are most likely to yield you the most potent compounds, the most tractable compounds, the best, the most stable compounds, and then thinking about using computers we're now using to design our synthetic routes, because I'd rather come up with my CDs not having 21 synthetic steps, but 8 synthetic steps, because it's a lot more cost-effective. The computers, I think, an AI-based design are really helping us now across biology, chemistry. They're helping us in the clinic. It's really helping us everywhere. And it's not about replacing scientists. It's about aiding scientists to make better decisions.
Yeah. So it's a man plus machines and
man plus machines. Exactly right. Man plus machines
and algorithms. So this is obviously a great way to look into the future. So with those learning and new technologies, particularly the learnings from the pandemic. So how do you see our industry, the biopharmaceutical innovation, evolving in the coming 10 years? So let's say 10 years from now, if I visit you in Cambridge, we have a chat, we should probably do that. So what will we talk about? What type of innovation will we thinking about at that time, in 10 years time?
So I think the AI-based design, AI in everything that we do, whether it's biology, chemistry, clinic, patient selection, I think that will be everywhere, right? We're going to have scientists that are comfortable with data and are working with data scientists in a way that basically becomes seamless. I think you're going to see a lot more gene therapies and cell therapies. I think in my area, in the non-oncology areas of AZ, I think you're going to start to see a lot more therapies that are going after reversal of disease or curing of disease, not just treatment of disease. So I think some of the things I'm seeing around CRISPR gene editing, base editing, cell therapies, I think is regenerative therapies is very, very interesting and exciting. I think, programmable therapies, we've seen it with the vaccines, with mRNAs and with adenoviral-based vectors, but it's true now, again, across disease areas, you're going to see many more programmable type therapies, which have been antisense oligonucleotides, siRNAs, mRNAs being applied across a broad set of diseases. And actually, I think mRNA is going to evolve to self-amplifying mRNA, which means lower dose, lower cost, and easier to do over for chronic use versus acute use in vaccines. And then I think we're also going to be using a lot more the way we conduct clinical studies, how we've had to adapt to a digital world during the pandemic is also changed a little bit how we all work, right? We wouldn't be doing this like this 2 years ago, but we'd also be running our clinical trials very differently. We'd be distributing our drug substance very differently to a site versus straight to a patient. So I think all of those types of things will continue to evolve, making us hope more efficient. And I hope ultimately our industry just becomes more efficient. I think, we're still, relatively unproductive. And so I think our industry needs to continue to improve. But it's a great time, I think, to be a scientist, because I think we're uncovering a lot of new disease understanding, a lot of new pathways, and there's capabilities now that can really have us starting to dream about cure and reversal of disease versus just treatment. It's not so far away, I think, in some areas.
So in another word, some of the chronic disease that you're working on could become even, be modified or as you described, even reversed.
I hope so. I think that's what we have a catchphrase that we use, which is called "Life Without Limits." We want to turn chronic illness, people with chronic illness, to be able to live their lives normally, not impaired or impeded in any way. And to do that, obviously you have to have good treatments and you can stop disease, but I think the real goal now is to see if we can reverse disease, regenerate tissue, and then even cure.
Thank you, Sir Mane. It's a great opportunity to catching up and hear your insight at the WuXi Healthcare Forum 2022.
Thank you, Stephen. Thank you for having me.